This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on September 11th, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. And I just realized, reading the date, this is a sad date in U.S. history. So remember all the people who died on this day many years ago. Today we are recording in Denmark. We are at the largest research university in the country. It is Aarhus University. Did I get that right? Aarhus, A-A-R-H-U-S. I practiced a lot with Maddy, so that's why I got it right. And I have four guests from the university. We're going to chat about the work that they do. It's a remarkable collection of virology and virologists here at the university. And by the way, looks cloudy today. What's the temperature? Do you know roughly? 14, 13 roughly?
Yeah, maybe.
So it's a typical cool weather that we have here, but at least it's not raining. Okay, so my guests here today from the Department of Biomedicine, Soren Paludan. Is that good?
That's perfect.
Welcome to TWiV.
Thank you.
We passed your eyeglass store this morning walking around. Paludan Optics. It's not yours?
Not really, no.
Maybe
I don't even get my contact lenses from there, so maybe I should.
Well, you should, because if you go in and they see your name, they might give you a discount.
They may, yeah.
And also from the Department of Biomedicine, Trina Mogensen. Welcome to TWiV.
Thank you.
Did I say your name correctly?
Yeah, this is fine.
Trina Mogensen. So I'm very sensitive about name pronunciation. How do you pronounce my name? Does anyone know? Say it.
Black and yellow.
Very good. I tell my students, black and yellow. Do you know this song, Black and Yellow? No? Yes? Okay. That's how you pronounce my name, Black and Yellow. Okay, from the Department of Clinical Medicine, Ole Søgaard, but I probably got it wrong. Welcome to TWiV.
Thank you very much. You actually got it fairly right.
Ole. So the first part is O-L-E, right? That's Ole?
Yeah, and in Danish it's Ole.
Ole, okay. And then what about the last name?
Søgaard.
Oh. Søgaard. Okay. Well, welcome to TWiV.
Thank you.
And finally, Madalina Carter-Timofte.
Yeah.
Is that how to say it?
Yeah.
We call you Maddy, right?
Yes, that's much easier.
Welcome to TWiV. And you are the reason I'm here, because you reached out. Actually, I emailed you and I said I was coming, so now I'm here because of you. So you take any blame.
Yeah.
So if you want to blame someone, you can blame her, okay?
Yeah. Thank you for coming.
We are all going to talk about virology here. So when you have the O with the slash, how do you say that?
U.
U? Okay. So I was just in Sweden and I got lessons on A with the circle, A with the dots. It's very complicated. All right. So let's talk first about a little bit about your history. I want to know where you're from, where you got educated, so forth. So are you from Denmark?
So I can say, yes, I'm from Denmark. I actually got my first education here at Aarhus University.
Right here,
or I think 200 meters down the road. I studied chemistry and biotechnology. Mm-hmm. And then I went to Oxford to complete my studies. That's where I got interested in immunology, microbiology and stuff. And I came back to do my PhD here. I went to the US, where I completed part of my PhD. I was at the Cleveland Clinic. And you'll hear soon that Trine went there too. And from there, I got back here to Aarhus, and at that point, I got the opportunity to start my own group. And from there, although quite early in my career, I started as independent with a focus on herpesviruses and how they interact with the immune system. And And I guess that's the focus of our work still.
Cleveland. I've been there.
Nice place.
So, do you remember how far back you wanted to be a scientist?
I don't know. I know my father's a priest. I know I did not want to be a priest.
You did not want to be
I did not want to be a priest.
Okay.
So I guess you could say the contrasting choice is to be a scientist. I don't know whether that was a driving force, but nevertheless, I think since I was in high school, I wanted to do something related at least to science.
Right. It's funny. My last guest, Erling Norby from Sweden, is a retired virologist. His father was also I don't think he was a priest, but he was in the church. So priests are one religion, but in other religions they call them something else, and I don't remember, but that's what he was. And I think I asked him the same question, and he had similar answer. The opposite of religion. Although science is our religion, right? It's the way we think the world goes. Trina, where are you from? Denmark, I guess, right?
I'm from Denmark as well, and I can beat Søren on a few points because I was actually born at this campus.
You were born in the hospital.
What is now this beautiful white building, the medical library, used to be a hospital of obstetrics, so I was born just down the street.
You feel a connection with it?
Well, I spent a lot of time because I also lived here in a dormitory while I was studying medicine.
So you went to medical school here as well?
Yeah, so I went to medical school, and already during medical school I was very interested in science, so I did a research year, which you can do, and became very interested also in basic research. I also did a year in Paris to study biochemistry and medicine, so I was all the time very drawn to both science and clinical medicine.
So do you think because you were born here you became a scientist?
No, not really.
What was the driving force, do you know?
Well, up until I was 15 years, I was actually more into literature and languages, but then I had this very interesting 1-week stage in the Department of Human Genetics in this university. I thought I would be a writer or journalist at the time, but the local newspaper didn't want to have me there for this week. So then I came to this Department of Human Genetics, and I just became very fascinated and interested in science. So this was in a molecular immunology lab working on transcription factors, gene expression, and that really caught my interest and became very decisive in choosing a career in natural sciences and medicine.
So I interrupted your academic career. So you did a 1-year fellowship after the MD, right?
Yes.
Where you got interested in research, right? And then where did Cleveland come in?
Yeah, my career is a bit unusual because then I went 1 year to Cleveland also as a medical student. Partly because I met Søren, and then because
You met him in Cleveland?
No. I met him before Cleveland, and then I did another research year in Cleveland, even though people told me this must be blind luck to go to Cleveland, this old industry city at the Great Lakes, only famous for the river that caught fire in the 1970s. So I had a very nice research stay in Cleveland at the Cleveland Clinic. So this was really a mecca for interferon research in the late 90s. So that's why we went there.
What's his name? Who's there?
I was with Brian Williams.
Brian Williams, right.
He was in the Department of Cancer Biology.
Is he the one who made the big ISG database?
Yes,
Oh, okay.
yes. He was very much involved in this and working in double-stranded RNA signaling.
And Ganesh Sen is
Exactly.
there. Was he there when you were there?
Yes, Ganesh Sen and Bob Silverman.
George Stark.
George Stark
was a big...
These were very strong in interferon biology, having been involved also in the identification of JAK-STAT signaling.
George Stark, right?
Yes.
Right, okay. I know all those
With
people.
Ian Kerr.
Yeah, so there's good science in Cleveland, despite its bad reputation. There's also Case Western, another good institution, right?
And the Cleveland Clinic is famous for its cardiology department.
Right. Floyd Loop was the famous cardiologist who worked there. I think he did heart transplants too, maybe? Was he a surgeon or just I don't remember. My father was a heart surgeon, and when he died, Floyd Loop talked at a memorial service for my father. So that's why I know. And his wife was head of the Red Cross.
Oh, okay.
What's her name? I should know. Floyd Loop's husband, wife. Bernadine Healy.
Okay.
Because she was also head of NIH for a while. That's why I know. So she was his wife. Yeah. Okay. I'm sorry to digress. We never digress usually, so I'm sorry to do that. It's a joke that nobody gets, but we digress. And after Cleveland, you came back here?
Yeah, then I came back and finished my medical studies, and from the work I did in Cleveland and before I finished my PhD. And then I went on to have clinical training specializing in infectious diseases, and all along did some research. And then at some point I received funding and got the opportunity to work part-time in the clinic and part-time in research 50-50, which I have now been doing for many years, and established a research group which is actually based at the hospital mainly, but also with strong connection to this department.
So you continue to see patients half-time?
Yes, and now I'm specialized very much in viral CNS infections and also in primary immune deficiencies. So I'm basically very interested in host-virus interaction and how immune deficiencies predispose to severe or rare invasive viral infections.
All right. Well, we'll talk about your science. Ole. Is that right? Ole?
Ole?
Ole.
Yeah, you got it.
Ole.
Yeah.
Where are you from?
I'm from a small city town about an hour's drive from here called Viborg. So not actually born here or lived here when I was younger, but I went to medical school here. So when I was about 4 years into medical school, I thought I needed a break from exams and studying. So I wasn't sure whether to go traveling or to do research. So I kind of wanted to do a research year, just like Trine mentioned that you can do here in Denmark, but at the same time I also want to travel. So I went to, a friend of mine was at the Department of Infectious Diseases doing this research year. And I went there and spoke to the head of the lab, Aarhus Søgaard, about this potential research year or something. And I also mentioned that I would like to travel. So he said, well, I have this project that you can do. You have to go to Australia for part of the project, but this is an opportunity to do a research year. So I figured, okay, I'm up for it. What do I have to do? So basically he told me, well, you have to study pneumonia in intensive care patients and the pathogens and also the treatment. So fine, I was up for it. So I started, I think, around September 1st that year. So it was in 2002. And after about 3 hours in the research department, he said, I think you better go to Australia straight away. I was like, oh, okay, I'll do that. So he called up a travel agency and said, when is your next available flight to Melbourne? I was like, okay. He said, well, it's pretty full for the next couple of weeks, but we do have one in exactly 1 week from now that is available. And he said, can you go in 1 week? I was like, yeah, I think. So he said, yeah, it's September 11th. Okay. So he hung up and I got the ticket and I was like, September 11th. So this was in 2002. So you just mentioned what happened in 2001, September 11th. So I guess that was kind of a test of character maybe. So I was on a half-empty plane to Melbourne, but I got there safely and I got back and it was fun. So I enjoyed that very much, doing research. So throughout medical school, I stayed connected to the department and I did some research on the side. And then I did my internship here at the University Hospital in Aarhus. And I realized that I wanted to do a PhD. So straight after I did a PhD in clinical epidemiology, also HIV related, which is what I'm doing now, but with large databases, this nationwide database where we can draw some healthy controls and compare comorbidities and things like that. So that was, it's pretty easy to work with because you have these very large data sets of high quality. And I also enjoyed that, but towards the end of my PhD, I did this randomized clinical trial with the pneumococcal vaccine and adjuvant, a TLR9 agonist, which was this investigational compound. And I really enjoyed the more translational part of science. And I just decided this was maybe the direction I would like to go with my career. And I went to the national or the annual HIV conference, which is called CROI conference. Yeah, you probably know it. And it was in 2010, and there was this first case of a patient that had been cured of HIV.
The German patient?
Exactly, the German patient, Timothy Ray Brown, who is actually American, as you know. And I was like, I want to do that. Work with HIV cures. I went back, and I have some great colleagues. Some are here today. Martin Tolstrup and Lars Ostergaard, who is now professor at the department. A brilliant PhD student we had at the time, Thomas Rasmussen, who's now in Melbourne, coincidentally. And we basically started this research within HIV cure with no basic knowledge to work on whatsoever, but we thought it sounded really interesting and intriguing. So that's what I've been doing part-time, then part-time clinics ever since.
So I would like to talk to you about HIV cure today as well. There's been, since Timothy, there was one other paper published of a cure, a bone marrow transplant-based cure, right?
Yes. Very recently, there was a publication of a
London
now
patient.
called the London patient. Yeah. And there's also a Düsseldorf patient who looks very similar to the London patient, but who's just lacking behind in his treatment interruption. But they also appear to be cured and are off treatment and doing well, as far as I understand.
Okay. Trina, I just realized why your name is familiar. You have a very good friend who used to write for the Chicago Tribune, Trina Suderos. Her family is actually, well, Suderos sounds Greek, but it might be her husband's name. Well, and Trina is a Nordic name, right?
I think it's just the nickname for Katrine in our country.
It's Katrina, huh? Okay.
Actually, in the US, people always call me Trini.
Trini? Well, I know how to say it because of Trina, who since left journalism, but she was a wonderful science writer. You know, it's always good when you find a good science writer, and she used to call me all the time and ask me, and she listened to the podcast, and so that's why. Okay, Maddy, tell us your history.
Yeah, so I was born in Romania, and I lived there for the first few years of my life. And then when I was 10 years old, my family actually decided to relocate to the United Kingdom. So I grew up in the United Kingdom and I studied there. I went to Lancaster University where I studied my Bachelor's of Science. And it was during this time that I had my first sort of taste of lab work. And so towards the end of my Bachelor's, I thought, this is really interesting. And I was very curious. But I wasn't sure if I was ready for the full PhD life. So what you can do in the UK, it's called a Masters of Research. And this is just a 1-year, very intensive research program where you have rotations in different labs. So I did this at the University of Liverpool, where I had the chance to work with malaria at the tropical school. And also I had my first taste of viruses because I had the chance to work with a respiratory syncytial virus. And then at the end of my master's, I thought, okay, this wasn't too bad. I think I can handle a PhD. So I looked online and I went on this website, very simply called findaphd.com. And the one thing I knew I wanted was to travel again, and I wanted to go to a different country. So I looked all over the place. There's great opportunities for anyone looking for a PhD in many countries on this website. And then I came across this PhD position in Tina's lab in Denmark. And I should say that my PhD is actually part of a larger group known as EDGE. And this is a European funded program. There's 15 virologists, 15 PhD students, all working towards understanding herpes viruses. So I applied for the position here in Denmark, and I was very lucky and fortunate, and I got this Marie Curie fellowship and ended up in Tina's lab 3 years ago.
Mm. So do you remember why you got interested in science?
So both my parents are actually scientists. So I remember from an early age having scientific dinner time conversations and there was always some funny samples in the fridge that we shouldn't touch. So I was always around science, but actually during my teenage rebellious years, I remember saying to my parents, I do not want to do what you're doing. I don't want to be a scientist. And I didn't want to be a PhD student. But I guess curiosity kind of got the best of me. And even though I didn't necessarily want to do science at the beginning, I just kind of followed what was interesting to me and ended up in science.
I think it's interesting that in some of the countries I visited, a lab advertises for a PhD student. So you have a position and you can hire a PhD student. Whereas in the US, it's never individual driven. It's always a department. We recruit PhD students, they come and then they decide where they're going to work.
Okay, yeah.
So it's an interesting difference. You know, in the end, I look at PhD students as temporary labor, right? Because that's what they are. They come, they do a project, they help the PI, then they go, and they're off on their own. So it's kind of a weird in-between kind of position. But when the PI hires someone, it's clearly you're working for me, as opposed to a PhD student in the US.
Yeah.
I think that's an interesting difference, right? Definitely. So that's the way it is here in Denmark, right? You advertise for PhD students. And I think in Sweden it's the same, as far as I could tell. I wonder how many other European, you have any idea if it's the case in other European countries?
I know it's the same in the UK. I think also Germany, perhaps.
Interesting.
Yeah.
Okay.
But we have different models. So the model that Maddy entered through is actually not the usual one. The usual one is that the PhD student submits an application
to a lab.
No, to a funding source, and that can be a department, or it can be a funding source, private or whatever. And then you get your position, but you normally have a sponsor in the lab. Meaning the PI.
Yeah.
And that's actually the normal route for a PhD in Denmark.
Okay. So you apply to a funding source first. That's interesting, because we don't do that at all in the US. In the US, there's a lot of controversy about PhDs and postdocs, because they do the work for the lab, but it's a temporary position, and then you leave, and then you get more, and then they leave. So there's this we call it a pyramid scheme, right? Where there's the PI at the top and lots of people below, but there's very little room at the top for students. And so there's a lot of discussion about what to do about that. The problem is PhD students need to be trained, but they also have to do work for the PI, so you get your grant renewed, right? So there's this confluence between training and doing research, and they really should be separated, and we can't figure out how to do that. Because students should be just trained and not have to produce work for the grant renewal. That should be done by a more permanent person. Anyway, I'm sorry to diverge into that. I didn't mean to. But let's talk about some science now. Soren, let's talk about sensing of viral DNA, which is what you work on. So I should tell you all, I read a few of your papers. I always do that in preparation so I can have a proper discussion, because afterwards people always say, wow, you read our papers. And I'm like, what do you think? I would just come in and not have read anything? But that's part of the fun of doing these. So we have talked a little bit about sensing of DNAs in cells. So let's talk about that. So you worked on some herpes viruses, herpes simplex virus. How is that viral DNA sensed, and where in the cell?
So I think it's debated.
Good.
I guess the current dogma is that herpes virus DNA is sensed mainly in the cytoplasm, which may be a bit counterintuitive for a virus that replicates in the nucleus. And I guess it should also be said that there are papers, very strong papers, including one from Akiko Iwasaki, where they actually show that it's mitochondrial DNA that leaks out of the mitochondria of an infected and stressed cell that is actually being sensed. So the DNA source being sensed is still debated.
That's interesting, because for dengue, that's been shown also.
Exactly, yeah.
An RNA virus damages the mitochondria, and then the DNA is sensed.
Yeah.
So that's plausible. But isn't there also some indication that there are nuclear sensors as well?
Absolutely. I mean, that's a very active field now in the cancer area that actually DNA damaged cells, stressed cells, cancer cells, they leak out DNA from the nucleus into micronuclei and other cytoplasmic temporary compartments. And that DNA, extranuclear DNA, is being sensed. Whether that happens during virus infections, it may or it may not.
But we know that herpes viruses are taken up by endocytosis, right? And then the capsid docks onto the nuclear pore and the DNA comes out through the portal, right? So where does the opportunity for cytoplasmic sensing arise?
So that's the counterintuitive part, I guess. And it should be said that there are papers actually indicating that some nuclear sensing does take place. However, what I think is maybe overlooked is that it's a relatively small percentage of the virus particles that enter into a cell that manage to make it to the nucleus. So it's not a safe journey for a virus to enter into a cell. So that's probably why so much cytoplasmic recognition takes place.
So you think most of the particles remain in the cytoplasm?
Yeah, I mean, we have some data indicating that most of the DNA ends up in the cytoplasm, actually.
It's not a very efficient process, right, for the virus anyway.
No, it's not, but I think that goes for many viruses. For influenza virus, some people say that it's only 1 in 1,000 particle that can really replicate and stuff. So I think if as a virus you can produce a lot of progeny, then it may not matter so much. And in fact, there are also some indications suggesting that some viruses actually have an interest in activating some immunity, not least for herpes viruses who want to establish latency. So you actually want to be stopped by the immune system.
Okay, so that's part of the viral strategy, to make lots and lots of particles and only a few actually succeed at infecting an next cell. Right? If they made a few particles, then they wouldn't get anywhere because they would all be eliminated probably.
Yeah, probably, yeah.
All right, so that makes sense that most of the capsids end up in the cytosol. So let's talk, what senses them in the cytosol? What senses the viral? Do they break open and leak viral DNA? Because are they coming in through endocytosis? Tell us what's going on.
So it differs between viruses and this question I think is studied in HIV and in herpes virus. In HIV, it seems that there are some cellular proteins that sort of nibble on the capsid structure, and that breaks it open. For herpes viruses, it may be the same, or it may be that the virus particle frequently just opens. Herpes virus particles are formed under pressure, so there's increased pressure in a herpes virus particle, and at the tip, sort of the cork of the champagne bottle sits. It's called VP6, I think, and that easily falls off, and then the DNA is just ejected.
Right.
So that probably happens quite often, and normally that's mediated by the nuclear pore. But it can happen in the cytoplasm, and one reason may be that actually many of the nuclear pore proteins, even though that's not well recognized, are actually also floating in the cytoplasm.
Okay.
So when the DNA gets there into the cytoplasm, then there are DNA sensors.
Yeah.
And several have been proposed. There's a long discussion, and you discussed one of our papers that Trine was leading a couple of years back. The DNA sensor that most people work on, and probably because it is a prime sensor in most cell types, is called cGAS.
Right.
We've
talked about cGAS.
Has received a lot of attention in past years.
Cyclic GAMP synthase, right?
Exactly. Yeah.
So it makes a funny cyclic. So does this protein bind DNA directly?
So it binds DNA directly, and that activates the enzymatic activity of this enzyme, and then makes the cyclic denucleotide,
right,
which is a second messenger for a signaling protein called STING that sits on the ER, and that's what starts the signaling. And that's a very complicated process, but the end product is production of the antiviral cytokine called the type 1 interferons.
Right.
And now we're back at what we discussed about Cleveland, interferon, and this is the main player in the early antiviral response.
So when STING is activated, it stimulates phosphorylation, right?
Yeah. So what it does?
What's the IR? The IRF?
RFs, yeah. So what it does after binding to the cGAM, the cyclic denucleotide, it will traffic from the ER to the Golgi, where a kinase called TBK1 is recruited.
Okay.
And that will lead to recruitment of IF3.
Okay.
And that's what eventually gets activated and drives the transcription of the interferon.
So IRF3 dimerizes, it goes in the nucleus and activates interferon transcription.
Yeah.
Right, and IRF3 can be activated by other sensors as well, or RNA sensors as well. Okay, and STING, what does it stand for again?
Stimulator of interferon genes.
Stimulator. So Dixon always asks us what these words stand for. Dixon is one of my co-hosts on TWiV. He's not a virologist, so he wants to know what all the words stand for. And mostly we use them without knowing what they stand for.
STING was identified by Glenn Barber. He's now in Florida, and he's a British scientist who's in the US. And I never asked him, but it may be that he's a Brit in the US, and STING made the song "I'm an Englishman in New York." So maybe that sort of stimulated the name. Who knows?
Well, so there's a connection with Dixon. Dixon wrote a book on indoor farming called The Vertical Farm, and STING had a quote on the back because he likes ecologically sound practices. Apparently, when you get wealthy, you become ecologically sound, right? So this whole idea that the portal is opened by this this is really interesting. I'm thinking of a champagne bottle being sabered, and the DNA is popping out. So any DNA in the cytoplasm is not good, right? The cell doesn't like that.
Yeah, the cell doesn't like that. It's okay with short stretches of DNA.
Right.
The longer the DNA, the more cGAS becomes activated.
Okay. Yeah. So you had a paper on that, right? Looking at the length of DNA that activates.
Yeah. So there's a clear length dependence, and that goes with the cells expressing a lot of exonucleases. So always continuously chops DNA, so it makes shorter and shorter stretches. So there's a sort of an on-off. There's accumulation of DNA, and there's removal by the exonucleases. Okay.
So what's the shortest DNA that will not activate STING?
So I know a 20-mer will not, a 43-mer will if you have enough of it.
So when we transfect cells with DNAs for production of proteins, that DNA is activating STING. Plasmids do, right?
Absolutely, yeah.
So is it better to have STING null cells to make protein?
Yeah, it is. And that's probably why everybody works on HEC2 and 3 T cells. Because they don't express cGAS and STING.
Oh. Yeah, that makes sense. Yeah. The other thing I learned recently is that if you use HDAC inhibitors, you get better expression as well. Okay. Because when plasmids get in the nucleus, they're immediately chromatinized and acetylated and silenced. And you only get a fraction of any DNA that you put in cells expressed. But if you use an HDAC inhibitor when you transfect, you will get much, much more. So remember that. If you're trying to make a protein by putting plasmids in cells, use an HDAC inhibitor and use 293. And then the cell will be full of your protein, right? So the DNA pops out of the capsid, sensed by STING, and then interferon is produced. So what allows the virus to replicate then, if all this interferon is produced?
So of course the virus has a lot of evasion proteins,
right? And
herpes viruses have a lot, as in a lot. So in the literature, there's now reported about 10 viral proteins that antagonize the cGAS-STING pathway. I mean, herpes virus protein. So I mean, if you want to be a herpes virus, you need to target this pathway.
So what you published on 152 in CMV, right, as an inhibitor of STING?
Yeah, I mean, I was not leading that study. That was together with Melanie Brinkman, where she basically shows that this inhibitor, it does not really block, but it sort of impedes. So STING, the trafficking I talked about before, occurs much more slower. And then what happens is then that sort of it gives the virus a head start, and then the interferon eventually comes along. We published a study where we showed that a herpes simplex viral protein called ICP27, which is actually an essential viral gene. It's normally in the nucleus and it's part of the viral replication machinery. It mediates transport of a viral RNA species. At a later time in the replication cycle, it traffics to the cytoplasm where it binds to STING. and it prevents recruitment of IF3 to the STING signal zone.
And then there are others you say as well.
Oh, yeah, others. Oh, yeah.
And there are also nuclear antagonists of sensing, right?
They're also nuclear, yeah. So there's one ICP0 which degrades IFI16. IFI16 is also proposed to be a DNA sensor. We were also involved in that work. IFI16 has several roles because it's also a restriction factor.
It's interferon-induced protein 16, right? IFI.
Yeah, exactly. Yeah.
So that's a sensor in the nucleus, right?
So there are papers on its sensing in the nucleus, but also in the cytoplasm. And a pool of IFI16 is in the cytoplasm, but the majority is in the nucleus.
So it can sense in the cytoplasm as well.
Yeah.
Okay. So you also mentioned that the interferon response may encourage latency. Is that right?
So there are some papers. I think they're mainly from the gamma herpesvirus literature, where they can actually show that the interferon response is essential for establishing a latent pool of viruses. And I guess it makes sense that the virus, since it wants to live, lifelong with the host basically, then why not take advantage of some of the cellular mechanisms to control replication?
Right. So maybe latency is one of the responses to interferon in a way, right?
Yeah, in a sense you could. Yeah.
And so when you get reactivation of a simplex infection, is that also being sensed by the same sensors?
So, there's not much literature on it. I think actually Trina and Maddy have some genetic data that links sensing to reactivation. Mechanistically, there's not much in the literature on that actually.
Because there you have nuclear DNA replication, right? So that should be sensed. But then the capsids are assembled in the nucleus and shipped out, but then there might be breakage on the way out too, right?
Yeah, and there may be, adjacent cells that sense a virus that is, it's produced during a reactivation, but I guess the neighboring cell does not know whether it's seeing a virus that's produced during primary infection or reactivation.
Right. So I was interested in this recent paper that you had, which described the sensing of incoming herpes simplex DNA in capsids, where you labeled the genome with click chemistry so that you could track it, right? And you find that, as you said before, the DNA is released from a fraction of the capsids, right? And that's what's being sensed by STING. And then the capsids are degraded also.
Yeah, so I guess at this stage, to be honest, we really don't know whether this capsid degradation, what's the importance of it, at least for the DNA sensing, but we can certainly see that the capsid gets degraded. At a certain point, we thought that it was actually the degradation that was driving the DNA release.
Yeah, right.
I'm not convinced. I'm not saying it's not the case, but I'm not convinced. So we'll need to dig more into that.
Yeah, you say that the release of the DNA is not proteasome dependent.
Yeah, exactly.
So the proteasome is degrading the capsid, but the DNA getting out is not a cause of that.
Yeah, so I guess one interpretation is that the DNA release occurs prior to the degradation of the viral capsid.
So another aspect of your work, which you do in collaboration with Trina, is looking at host genetics and susceptibility, right? And I know that we've also talked on TWiV about mutations in sensors that predispose kids to encephalitis, herpes encephalitis, from Casanova, and I think you collaborate with the Casanova lab, right? Do you ever find mutations in the sensing pathway that predispose kids to more serious herpes virus infections?
So, this is not work that we are driving. I think Trine is driving, and Casanova and other people. But we have been involved in one study, not looking at single genes causing disease, but more at population level, looking at SNPs. Yep. We have found mutation in IFI16. And we found one SNP that predisposes to severe genital herpes.
Right.
So
in 16, IFI 16.
IFI 16, sorry.
Okay.
Yeah. So that could indicate that this pathway, the STING pathway, since IFI 16 signaling is dependent on STING, so that could indicate that the STING pathway is important for some viral infections.
Right. But no polymorphisms in STING itself, the STING gene, or there are.
There are several. I mean, some of them are very frequent actually. So one allele is called the HAQ allele because the substitution in STING has the HAQ residues is as frequent as I think it's 20% in the American population.
It's not a null allele though.
That's debated, exactly. So when you overexpress it in HEC cells, you can actually see it being a null allele. However, I guess, we have some data, and I've also heard other people where if you co-express it with cGAS, it's a little bit more complex. It may be a hypoactive allele rather than a null allele.
Hypoactive?
Hypo.
Okay, so it's less, what aspect of it is reduced?
So the activation, so the signaling capacity is reduced.
Not the binding of the
cGAS binding, as far as I know, is not affected. It's more at the level of trafficking and recruitment of cofactors and stuff.
How about cGAS itself? Are there polymorphisms there that affect infection? It's interesting that I always wonder why there isn't more redundancy in some of these pathways, right?
Yeah, there probably is. So
but kids get sick when they have these polymorphisms, right? They get serious disease. So
yeah, that's correct. I thought you were asking about the cGAS-STING pathway. So here, I think until we have seen a human being with a loss of function mutation that cause a disease or causative for a disease, I think we don't know whether this pathway is redundant in human beings.
What about in mice?
In mice, it's essential in many models. Of course, all models are wrong.
And some are informative, right?
So we can certainly reveal very, very strong phenotypes in cGAS-STING knockout mice
with herpes infections,
herpes viruses, and also some bacterial infections. So clearly, and also I think you can malaria and also some intercellular bacteria. I think TB actually. So clearly, you can reveal experimental situations where this pathway is essential. Mm-hmm. In the real world, we don't know.
So is cGAS-STING the main cytosolic DNA sensing pathway? Is there anything else? You said IFI 16 comes out into the cytosol, right? So that is there anything else?
So I guess yes, there is. Well, so that's a long history. To make it very short. So first, if we talk about interferon and non-interferon, for the non-interferon part here, inflammasomes like IL-1, then cGAS is not a main player. That's probably AIM2, another DNA sensor. For the interferon part, cGAS is the main. There are others. Some of them are probably cofactors for cGAS, like IFI 16 is now understood to be probably a cofactor. And then there are others too. I think Trina and Maddy would like to talk about some of that, I guess.
Okay. I'll take that as a transition then. So Trina, you may know we discussed your paper on TWiV 456, and so we are now about 100 episodes away from that. This idea that mutations in the Pol3 genes.
Yes, I heard it. Maddy.
She showed it to you. Yeah.
Yes.
Yeah. I thought that was really very interesting. That a DNA virus is sensed by RNA, right? In the dengue story is a similar thing. An RNA virus is sensed by DNA sensor, the mitochondrial DNA leakage. So that's pretty cool. Now, Maddy, you were involved in that work?
The initial paper, not so much. I've been doing some follow-up studies, but a lot of my project was based on that first paper, yes, where they identified mutations in children. So it was upon primary infection with the Varicella-zoster virus, right, who then went on to develop very severe infection, central nervous system complications, or pneumonitis, and then whole exome sequencing was carried out, and they identified mutations in different subunits of RNA polymerase 3.
And nowhere else in the genome, or just Pol3 mutations?
Yeah, I think as far as I remember for that paper, so we look for very rare single nucleotide variants because the phenotype is also extremely rare. So often you should find only a few candidates and then try and functionally test these out.
In fact, based on previous literature where the TLR3 pathway was shown to be essential in herpes simplex encephalitis,
Right.
another alpha herpes virus, we anticipated more or less to find mutations in the TLR3 pathway because these viruses are pretty similar. So in collaboration with Casanova, who sequenced more than 200 children with herpes simplex encephalitis, we looked all over and actually did not find TLR3 defects in the VZV encephalitis cohort.
Interesting.
So this is something that we find is interesting and not fully resolved. But I would strongly argue that the Pol3 is important in some cell types in DNA sensing. We looked into expression levels in healthy individuals and found that cGAS is not very highly expressed in PBMCs. And since VZV infects monocyte T cells early during infection, we think that Pol3 proteins, which are highly expressed in these cells, probably play an essential role there. So this may reveal lack of redundancy, and this is maybe why we see a clinical phenotype. So this is why I think it sometimes is important to study humans, because there are differences between murine immunology and human immunology. So what we hope is by studying the patients that we get a kind of knockout to look into the role and impact of a given molecule in patients in human immunology.
Well, the cool part is you can see the mutations in humans. Then you can take cells sometimes and study those. You can reintroduce wild-type sequences to restore the phenotype and so forth. And then if you want, you can go in mice and see if you get similar findings, right?
Exactly.
So the Pol3 mutations are important in PBMCs. Is that right?
Probably also in other cell types, but at least we saw very low cGAS expression levels. So this might be one thing. Another thing is that Pol3 is sensing AT-rich DNA, and this is particularly abundant on several spots in the VZV genome. So this may be part of the specificity why defects in the Pol3 sensor may predispose to VZV infection as opposed to HSV.
Right. So for HSV, the main sensor appears to be TLR3, and Pol3 sensing doesn't occur in HSV?
It probably does to some extent. We looked into the VZV deficient children, and they had generally pretty normal responses in terms of interferon production in response to HSV 1 and 2.
So you can take cells from them and infect them, right? That's how you would do that experiment?
Yes, that's what we do in PBMCs, sometimes in fibroblasts, and now we're also trying to expand the model to look into a more physiologically relevant cell type. So we are introducing mutations identified in patients into neuronal cell lines to study viral replication and interferon production in neurons.
Do any other DNA viruses get sensed through this Pol3 mechanism?
I think adenovirus as well. Yeah. And there's also some evidence for EBV, because EBV, during latency, it shuts down transcription except for these small non-coding RNAs known as EBNA, I think.
Yeah.
And these seem to be transcribed by Pol3 in the nucleus. And there is also some indirect link to Pol3 sensing EBV. And I think also Legionella, if I'm not wrong, maybe has also been associated to be sensed by Pol3.
So what cell RNAs are transcribed by Pol3?
So it's mainly small non-coding RNAs, right? 5S ribosomal RNAs, some tRNAs. So I think there are no homozygous null mutations in mice or in humans because it has this essential housekeeping function.
I'm just wondering why those RNAs aren't sensed in the cell.
So what happens is when you transcribe these housekeeping RNAs in the nucleus, they then undergo 5' capping and they then exit the nucleus. So because they have this 5' cap, they're no longer sensed by the cytosolic DNA sensors. Whereas VZV, this is transcribed by cytosolic Pol3, and because this happens in the cytosol, there is no 5' cap, so you have this naked 5' triphosphate, and this acts as a ligand for Reg-I and downstream interferon signaling.
But the Pol3 transcripts are not capped, are they?
I think they are.
The tRNA and the ribosomal RNAs, I don't think so. It may be that they're sequestered.
Yeah.
I don't think that, does anyone know, are Pol3 transcripts capped? I don't
think they
are, yeah. So I think there's some other reason why they're not sensed. The tRNA may be that it's hidden in the fold, right? And the same with the ribosomal RNA, may be buried in the structure and so forth.
Yeah.
But certainly for capped RNAs, that's a reason that they don't get sensed. So are you still working on this project, Maddy?
Yes. So we then went on and looked into adults. And there we recruited adults with severe central nervous system complications. And we had 8 adults, and here in 2 out of the 8, we also found mutations in different subunits of Pol3. So we tried to characterize these in patient PBMCs. And now what we also try to do is knock out the function of Pol3 in a heterozygous manner, neuronal cell line, and try and study the role of these mutations in different cell lines that we cannot get from the patient.
So all these individuals with severe disease, none of them are vaccinated, right?
No, not the ones that we recruited.
So that's a good reason to get chickenpox vaccine, right?
Is definitely. You should always get the vaccine.
So here in Denmark, do people generally get vaccinated? No?
It's not part of the official vaccination program, and it's an ongoing discussion. Of course, there's economics, and there's also this back and forth whether the vaccine strain will give you lifelong protection and whether we will see more herpes-zoster in the elderly generation if we don't have endogenous reactivation, and also if we don't have exogenous exposure. So I think the increasing burden of Varicella-zoster in the elderly and immune suppressed has not been demonstrated to be due to vaccination in countries where they vaccinate, like the US and in Germany. So I don't think there's much evidence to have this concern, but anyway, it's a theoretical concern and discussion. And I think there are divergent opinion on this, but I think many clinicians and virologists would agree that vaccination is probably a pretty good idea.
Also, if you're older and haven't been, you should get the shingles or the zoster vaccine, right?
Yes, and there's this new subunit vaccine which appears to be very efficient and very safe. So we encourage this in patients.
Is that available here in Denmark?
No, not really. It's just not introduced by the company, although it's fully legal. So we are licensed. So we managed to get just 8 vaccines for some of these patients that I see in the clinic that have very severe zoster or very severe reactivation in the CNS. So we vaccinated a few ones, but it's not so easy to get the vaccine. But I think that will change soon.
Well, in the US, it's in short supply. In fact, you can go to a drugstore and they won't have it. You have to get on a waiting list. There's a lot of people want it because it's gotten good press, older people mostly. But if you go, you can't get it because they don't have any. They can't make enough. So, but I in the clinical trials, it had great efficacy.
Absolutely.
It was amazing.
Hopefully it will be more widely available.
It should be. I mean, this is a reason why when one company has a monopoly, it's a problem for health, for products that are important for health, right? If you can't make enough, you need to do something about it. Anyway, so I wanted to talk about your polio study, because this is a study that I've been saying for years should have been done. I've been talking for 20 years now that, the rarity of paralytic disease is an interesting problem, but also as well, the vaccine-associated disease is even rarer. So anyway, tell us, so you published this paper on looking at polymorphisms in people with polio. So tell us what motivated you to do that.
Yes, so I would love to say that I got this brilliant idea, but the truth is that a medical doctor, senior doctor, who is chairman of the organization for polio survivors in Denmark, read about our studies on herpes, encephalitis, and genetics, and actually suggested, so wouldn't it be an idea to look into polio virus or polio myelitis survivors? And like you say, the idea is that, like in herpes simplex virus infection, 80% of individuals are seropositive, so why do we only see herpes encephalitis in 1 in 250,000? Very rare. This can be stochastic, but we think it's largely determined by host immunity. So the idea is the same. During poliovirus infection, less than 1.5% will actually get CNS invasion and myelitis. The strain seems to be very similar during an epidemic, so virulence should not differ that much. So could it be host immunity that plays a role here? So this is how we got into contact with this polio survivor cohort, and we were also aware that we had to do this study soon because these are the survivors that got polio as infants or children in the large epidemics in the 50s, early 50s.
Getting old. Yeah.
So these are elderly people now.
And this is a Danish population, right?
This is an entirely Danish population. So we recruited 18 of those surviving, also trying to select those who were very severely ill and had major sequelae. So the whole discussion about whether these had post-polio symptoms, infection, we didn't go too much into. Basically, our material is not big enough to distinguish subgroups here. So these are just patients who had very severe poliomyelitis.
Do they have recurring paralysis, or was it all resolved in most of them?
Some of them have recurrent paralysis and probably is in this subcategory, and whether there's live virus still, etc., etc. But we have not addressed this issue. So we basically just did our approach doing whole exome sequencing.
From blood, right?
From PBMCs from patients. And then we looked into very largely all pattern recognition receptors and sensing pathways that might be implicated in poliovirus antiviral immunity. So in the literature, there's not very much, and because polio is a human pathogen, although with a recent mouse model, there's really not very much basic information. So the RNA sensor MDA5 has been implicated in recognition of poliovirus and interferon responses, but otherwise not so much.
Well, actually, that's in cell culture, right? In mice, it doesn't make any difference. It's
so
TLR3.
So we had to go very open-minded into this venture, looking at different things.
Can I ask you a question first? So I mentioned this years ago, this idea of paralysis is 1 in 100, 1 in 200 infections. And they said that's not rare enough to find a polymorphism. Like, obviously, you did find some. Because the herpes encephalitis is 1 in 250,000, right? So there you can get a better number.
Yeah, that's true.
But so you can get a polymorphism with 1 in 100?
I wouldn't call it polymorphism because they should be present in
many people.
2% of population. So we're actually looking at very rare, potentially disease-causing genes here. I don't think host genes can explain everything, but we think it may play a role. So this paper is the very first study we did in polio. We have something more recent on the way. But basically what we found was potentially disease-causing genetic variants in several cellular pathways. So one is in interferon induction, another is in autophagy. So we have more evidence to also suggest that defects in autophagy may predispose to this. And we also found some other death signaling pathways. So this is a very descriptive study where we have looked very much on the genetics and just did very simple studies infecting PBMCs with poliovirus and with the MDA5 ligands to look at interferon responses.
So you didn't have a control population, or you did?
When we do genetics, we don't sequence healthy individuals. We're actually not allowed to do this ethically because unexpected variants may appear. So we can only have a medical reasons for doing whole exome sequencing. So what we do is we compare to publicly available databases where 100,000 Caucasians or other ethnic populations have been sequenced. So we all the time compare with the frequency of these variants in the healthy population.
So the variants that you picked up in this study were not present or were present at a low level in the...
They were very rare. They were selected based on being less than 0.01%. So they're very rare, and they may play a role in some patients. We don't have a genetic etiology in the whole cohort. And I think that when you do genetic studies, it's also very important to do functional validation. So this is a very descriptive study suggesting some variants, and I think it needs to be followed up. And actually we did this and have now submitted a work looking more into a few patients with autophagy defects, where we can functionally show that these patient cells have more virus, more cell death.
So the patients with these polymorphisms, when you infect cells, they produce more virus.
There's much more virus replication, there's more cell death, and we can see less autophagy. So autophagy, we think, might be an antiviral mechanism here.
So do these cells have a defect in induction of autophagy after infection? Yes?
Yes.
Because, autophagy can be proviral for polio also.
It can. It's a very complex issue. Right. So it can go both ways. And actually in the literature there are more reports on a proviral role where poliovirus uses autophagy membranes for its replication.
Right.
So we're up against some evidence. But I think in some settings it might also be antiviral. There's also evidence for this.
So obviously if it's proviral and you reduce autophagy, you should have less virus. But what you find is you get more virus. How much more virus do you get? 10-fold, 100-fold?
100 to 1,000-fold, 2 to 3 logs.
So this is in PBMCs? Or
yes.
Kind of, yes. So did you look in neural cells at all?
We looked also in neuroblastoma cells,
SKNSH.
Yes, this one. And we actually introduced the autophagy mutation present in the patient in this cell line. And most of the results are based also on these neuronal cells that we think represent better the true scenario.
Well, I would guess that the mechanism is that you get peripheral higher titers and that leads to CNS invasion.
Yes, that could very well be.
Right, and it doesn't matter what's in the CNS because once the virus gets there, it's a problem.
It's overwhelming.
So it's just a matter of getting in, and so it makes perfect sense if these patients make a lot of peripheral virus that's the mechanism. You might want to look at induced human neurons from stem cells, from pluripotent stem cells. They're better than the neuroblastomas because we use those. The induced neurons are real neurons. These neuroblastomas, a lot of people use, but
in fact, we are through collaborations with people at Hannover Medical School trying to set up the system, and they have it very well. But then again, to do CRISPR-Cas, introducing the variant from the patient in induced pluripotent stem cells is doable but difficult.
Yeah, it can be done. Yeah, you can put it into the precursor cells.
Yes.
And then induce them, yeah.
Or even develop iPSCs based on patient fibroblast. That's probably a better possibility.
So one of the issues here is we don't really know what cells the virus replicates in besides the gut and the CNS, right? So is it just the gut that gives you most of the viral viremia? It may be. So you might want to look in gut cells. You know, there are plenty of like CACO cells to see if these mutations give you more virus there. Because you can imagine that, you get 100-fold more virus in a gut, you're going to get more viremia, and that could lead to CNS invasion.
That's very possible. Also in fibroblast, which we had available from the patients, we saw much more virus.
So there are many interesting things about this, because I've been thinking about polio my whole life, basically. Not my whole life, just when I was 30 on, postdoc on. So in mice, the propensity to get into the neurons and be transmitted to the CNS depends on fast axonal transport. So if you have a muscle injury that causes accelerated axonal transport, more likely to bring virus to the CNS. This is Julie Pfeiffer's work. And interferon. If you take away interferon, like IFNAR mice, you get more mice getting CNS invasion. So I'm surprised you didn't pick up interferon polymorphisms.
So was I, because I work in the interferon system and I hope to find something that we're used to work with.
Sure.
We did have less interferon in some patients, but we didn't have a significantly decreased interferon production when pooling the 18. So it's not a general interferon defect, but in individual patients, there were a few with interferon defects. So I wouldn't rule it out, but it was not the general overwhelming picture for all patients.
Well, it's only 18 patients. You can imagine that if you look more, you could find in other patients.
I'm sure in a fraction, interferon would also be important, but it was not a common picture.
I bet if you look in vaccine-associated polio, you'll find interferon SNPs.
Yes.
So, that the frequency is 1 in 1.5 million. Kids get the polio vaccine, they get paralyzed, right? The vaccine is fine. It's nothing wrong with it. It's definitely a SNP, and I'll bet you find interferon. That's what I've always argued. But are you going to do that? Do you plan to look at VAP, vaccine-associated paralytic
I would love to do this, but these patients are not very easy to identify and
Well, the CDC has a whole list of them, I can tell you, because I was involved in some litigation. A lot of these people sue the vaccine manufacturers when they get paralyzed, even though it's nothing to do with the vaccine. It's their genetics, right? But we can't prove anything. So the CDC has a whole list of all the VAP cases in the US.
That would be very interesting because for me, polio is a novel area, so we're only learning things.
Yeah.
And we have an obstacle because the rules here and probably all over the world are changing in how you can work with live poliovirus. I was already checked by bioterrorism and also since the WHO is trying to eradicate polio.
Yeah, sure.
And the oral live vaccine is getting stopped. Maybe we're facing even more rigorous rules about how to work with it in the BSL-3 facility.
Well, the oral vaccine is still being used, and in fact, most cases of polio globally now are caused by the vaccine, right? Not
so
by
that's also
so you could
a reason why it would be maybe nice to not use it.
I agree, but in the meantime, there are those patients, if you can find them. They're unfortunately in places like Syria where you're probably not going to be able to get them, right? Not going to get any blood from them. But it happens in Pakistan, which would also be tough, and Nigeria. So they still exist. But I agree that we should stop using that vaccine. But it's so nice to give. It's just you drink it, and it's easy to administer. It's cheap. So it's hard to switch to an injected. What was the other thing? Oh, so now there's another virus related to polio that's causing paralysis in kids. Enterovirus 68.
Yes.
Are you going to look at SNPs in those kids?
We thought of it because it's more easy to work with. So if we can get some of these children enrolled in a study, I would be very interested in looking into this.
I mean, there are people in the U.S. who are doing that also because we have I think up to 500 cases so far since 2014 of paralysis. It's much rarer than polio. It's way rarer than 1 in 100 or 1 in 200. So I'll bet there's a genetic component. And there have been outbreaks in Europe. Have you had some in Denmark? I don't know.
We had small ones, a few patients, but it's, I think, quite some in Japan and East. So it would be very difficult to collect a cohort. But it would be interesting.
I think that's worth
But mainly I'm
in
in the herpesviral CNS infection field, so polio was just
a side project,
interesting opportunity that came up, and then now we would like to expand further on it.
No, I think it's great, and I'm going to put it in my lectures now because I think it illustrates that. One thing I wanted to ask you, of all the polymorphisms you found, does each patient have only one or do they have multiple?
Most of them have only one. So we try to filter out these that are most likely to be disease-causing. So most patients we nail down to one. But again, as long as it's very descriptive and we cannot really reconstitute patient cells, I wouldn't say they are proven. So these are candidates suggested for further studies.
So does any one mutation occur in more than one patient?
We actually have one in the autophagy pathway, and that's what we're trying to publish now, where we have this, the same gene, not the same location in the gene, but anyway, the same gene affected. Yeah.
Well, I think it's quite interesting. I hope it encourages other people with cohorts to do the similar things. It's easy to get blood and do the exome.
Yes, and like I said, this has to be done within a few years, probably, in order to have still surviving, at least in the Western world.
Yeah, but you still have kids with vaccine-associated polio. They're much younger, so they're candidates. You can reach out to them.
Yes.
You know, I found with social media, you can reach out to patients now very easily. So we did a crowdfunding for Entro-68 research, and you can find every parent whose child has been paralyzed. They're on Facebook, and they're happy to help. I mean, I'm amazed that they're so willing to help and support your research. So you can find them. You have to use social media.
That is also my experience, that people are very interested, even if we tell them this is not going to change anything in your specific treatment or your family, but we can gain insight into this disease, and people are very willing to participate, even if it's quite basic research.
And that's exemplified in HIV/AIDS. Patients are really willing to participate in trials of all kinds, right? Even though it may not help them. It's remarkable. Exactly. So one last question. You've looked at polymorphisms for polio, for herpes viruses, for influenza we didn't get to talk about, but what can we do with this? We can understand basic mechanisms, but can we help patients in some way?
That's a good question. That's our hope, although I think the basic pathogenesis understanding justifies these studies. But for instance, for influenza, we found individual patients with defects in interferon production, again in the transcription factors, IF3, IF7 that we mentioned previously. And of course, one conclusion here would be these are very rare variants, very disease-causing, almost no interferon in the patients. To go out in larger cohorts of patients with influenza or very severe influenza and look whether these are present as polymorphisms, so more frequent, maybe not as deleterious. And can we then better identify individuals who should be vaccinated, who should receive prophylaxis, testing their families. So of course we hope to have this aspect when we are very certain that there's an impact of it, to have genetic counseling, prophylaxis, early treatment. This is always the hope.
Maybe we can get more people to vaccinate when we say, oh, you have a polymorphism that's associated with severe flu, right?
Yeah, that's one thing.
Of course,
for the herpes encephalitis, it has been discussed for several years now whether type 1 interferon could be an adjunctive therapy to acyclovir, since we have acyclovir only since the 1980s, and the mortality is still 20-25% with major neurological sequelae. So maybe if we really can establish that type 1 interferon in the CNS is so essential, it might be something that could be done. And I know there are clinical trials looking into this.
How is vaccine uptake in general in Denmark? Is it pretty good?
It's generally good, I think, compared to other countries, but we have the same issues with the sometimes decreasing uptake for measles.
Right.
But generally it has been very good historically.
Okay. Let's talk about HIV/AIDS cure, Ole. I wanted to have a little historical background of the disease in Denmark. When was it first recognized in this country?
Sure. I think it was first recognized in Denmark a year or two after it was recognized in the US. So in the US, there was this case report of 5 homosexual individuals with pneumocystis pneumonia in 1981.
Right.
And I think a year or two later, there were the first patients that were identified here in Denmark. And actually, there's this case report in I think the Lancet, where a Danish female surgeon had been to Africa in the 1970s and had this symptom complex and died of severe immune deficiency a few years later. And it was actually traced back that she was HIV infected and had become infected while operating in Africa in the 1970s. So it has been known for a while and has been well characterized here too. So I think that the epidemic more or less progressed in the 1980s and 1990s similarly to the rest of the Western world and the US.
Okay, so what's the current situation? Do infections continue to occur? How many people are living with HIV/AIDS in Denmark?
Of course, so now it's a treatable disease that you live with and not necessarily die of. So I think the annual rate of infections is around 200 new infections per year and has been quite stable for the past 10, 15 years or so. It's probably declining slightly, but it's not like it's a major drop off. It's just maybe 2 to 20 cases less per year that we see gradually declining.
What populations are these cases occurring in?
So about 50% of the HIV-infected individuals in Denmark are homosexual men who have sex with men, and about, I would say, 25% are probably immigrants from Africa and Southeast Asia. And the last quarter is just
IV drug use?
Not many IV drug users, more just native Danish men and women who have become infected here.
And what's the size of the population living on antiretrovirals?
So it's approximately 5,000 individuals now, 5,000 to 6,000 individuals in Denmark. So it's somewhat, the prevalence of HIV infection is somewhat less than in the US, but I think it's around, around 1 in 1,000 that are infected and live with HIV in Denmark.
Okay. All right, so let's talk about what we call a cure. So you have a person who's infected and living on antiretrovirals. What do you mean by a cure?
Yeah, that's an excellent question. So I think there are two concepts of a cure. So one is a sterilizing or a complete eradication of the virus, basically having an infected individual that is completely freed of the virus. So there's no virus, or at least no replication competent virus left in that individual. So that would be a similar situation to an acute bacterial infection where you treat with antibiotics and the infection goes away and is completely resolved. That looks to be the case in the patient that we discussed in the beginning, the Berlin patient, Timothy Ray Brown, and probably also in the other two patients, the London patient and Düsseldorf patient, where they went through a procedure because they had cancer. They needed a stem cell transplant to be treated for the cancer, and in this process, donors were found that had this mutation for the HV co-receptor CCR5. So they were homozygous for deletion in CCR5. So they didn't express the co-receptor for HIV. And once the patients received this new immune system from these donors, they more or less became resistant to the virus. And we think that is how they were cured. Possibly a graft versus host reaction also played a role in this whole concept, but we think that's what occurred in these patients. So that is one concept, so a sterilizing cure. I think what we've come to realize over the past 5 to 10 years, it's really hard to get rid of HIV because it's retrovirus, so it sits in our genome, and many of these cells that are infected are latently infected, are not very transcriptionally active. So to remove this HV DNA copy that is within our HV genome, within our own genome, it's really difficult. So another concept is a functional cure, where there might still be some virus left, still some even still some replication ongoing, but the immune system is able to control the virus. So treatment will not be needed. The immune system can keep the infection in check, and there's no disease progression. And I think also very importantly, there's no transmission of the virus to other individuals, because if there would still be transmission, I don't think it would really be a cure. It maybe for the individual, it might be good, but not for the society.
Okay, so this I know is not an easily answered question, but what are the cells where the latent DNA is? There must be some bone marrow cells, right?
Yes, so we think so. It has been found in bone marrow. I think the generally most well-characterized reservoir in terms of cell types are CD4 T memory cells.
These are long-lived, right?
Exactly. They are very long-lived central memory cells and effector memory cells. Also, T stem cells have been shown to be infected. And these cells are very long-lived, so have a lifespan of years. And during their lifetime, they can become activated, so they can start producing virus again, or the virus can start to replicate in these cells. But they can also go through homeostatic proliferation, so they can actually proliferate these cells. So this is also one of the mechanisms that we believe maintains the reservoir, is that some of these infected T cell clones actually expand over time and then contracts again, just as a natural part of T cell homeostasis.
So these cells reside in many places: bone marrow, peripheral blood, tissue resident, right? They're
Yes.
all over.
We think they're all over, and they have been found all over, more or less.
All right.
So,
you know, a cynical question would be, why not, you know, eventually everybody with AIDS is going to die, and, you know, they can live long lives on antiretrovirals, but why not just prevent infection rather than worry about curing them?
I think preventing infection is at least as important as curing infection. So, I think it's a multi-layered question. So there's some benefit to society of not having to treat people for 50 or 60 years with daily antiretrovirals. So there's some economical benefit for the society. There's also the component that not everyone who's infected gets treatment. So in most countries, that number would be 10 to 20%. In Africa, it's probably much more than that are infected but not on treatment. So they might not be aware that they're infected. So these patients will probably continue to spread the infection because they're not on treatment. For the individuals, I think some patients are perfectly happy just taking one pill a day and not really worrying about the infection. So they live a completely normal life, have jobs, families, whatever, and are not really affected in their daily activities of the infection. But there are also a lot of individuals who are really bothered by the infection, having to take daily medication for the rest of their life. Also with the potential side effects of these medications that might have some effects on their kidney function or other organ functions. So there's for these individuals a strong drive to really get rid of the infection and stop taking daily medication. And then there's a whole stigma around being HIV infected, which I know bothers a lot of individuals. So this is something that should have been eliminated by now, but still exists. So there's still a stigma about being HIV infected, at least in some societies. So this is also, I know, a powerful motivation for some individuals to get rid of the infection.
So let's talk about some of the approaches. You've tried some, and you wrote this nice review article, Clinical Interventions in HIV Cure Research, where you talk about all the ones that have been tried. And then at the end, you mention immune-based therapies, which you talked about. Functional cures, right?
Yeah.
And this includes toll-like receptor agonists. And you've done a clinical trial with one, the TLR9 agonist that you mentioned.
Yeah.
So tell us about that.
Yeah, sure. So we at least think that by improving immunity in these patients, we might be able to help the immune system control the virus infection. So we've been looking for various strategies to really improve immunity in these patients. So one strategy would be to maybe develop a vaccine that would enhance HIV-specific immunity. But we want to test just the concept of some broad activation of innate immunity. And TLR agonists have been developed for this purpose, and TLR4 is used as an adjuvant with the human papilloma virus vaccine. So we know these have beneficial effects for the immune system, and administered alone, some of these TLR agonists really enhance NK activity and the cytotoxicity of NK cells, but also of CD8 T cells. So we were interested in just doing a pilot study where we administered a TLR9 agonist, which is not a licensed molecule. We got this molecule from a small German biotech company. And we asked patients to take this drug for 4 weeks. And during these 4 weeks, we took blood samples before, during, and after treatment, and basically measured their T cell responses, their NK cell responses, also activation of dendritic cells and other cell types. And what we saw in these patients was a very strong type 1 interference response. This was very evident in peripheral blood. We also took sigmoid biopsies from these patients, did RNA-seq, and saw very strong upregulation of these RNA transcripts for type 1 interferon regulated genes. Interestingly, the patients didn't experience any symptoms in the or side effects in the form of flu-like symptoms, which you might expect. We think this is because this drug gives a very clean type 1 interferon response and not so much TNF alpha induction or other cytokines that might really cause systemic side effects. So it was very well tolerated, this drug. We saw the immunological effects that we're hoping to see, so activation of NK cells, T cells, and also they seemed ex vivo to have enhanced killing effects. So this was really the goal that we're hoping to see.
These were in non-HIV infected patients, right?
This
Normals.
was done in HIV infected patients, but on treatment with completely suppressed viremia. So they were doing well. So it's basically well-treated patients that were taking their daily medication. So we expanded this a bit because we saw these immunological effects, and then we speculated that if we administered this drug for more than 4 weeks, maybe for half a year, we might actually be able to reduce the size of the reservoir, because we hypothesized that by boosting immunity and having a potent immune response around, maybe this eventually would kind of chip off the reservoir, or kind of eliminate the cells that became activated over time. So we did this for half a year in patients that volunteered to participate, but we didn't see the effect that we were hoping for. So we saw all the immunological effects that we had seen in the first pilot study, but we didn't really see a reduction in the reservoir. And we asked some of these patients if they were willing to interrupt their treatment as kind of a check of if something had changed in the infection. And what we observed was that all these patients rebounded. So there was one individual that almost went for half a year without rebound, but then eventually rebounded. So clearly this was a good strategy to boost immunity, but it didn't really produce a functional cure.
TLR9 is a DNA sensor, is that right? Yeah. And so you use molecules that are mimicking DNA.
Exactly. That binds to and activates the receptor.
So how do you measure the reservoir, by the way, the latent reservoir?
Yeah. There are multiple assays to measure the latent reservoir. So in our lab, we have established several of these. So there are the PCR-based assays where we just measure either HV DNA.
From peripheral blood?
Exactly, from cells from peripheral blood. Then we enrich for CD4 T cells, and then we measure how many copies of HV DNA there are in say, 1 million CD4 T cells.
So the peripheral blood is a good surrogate for the whole reservoir?
So we also tested that in a different study, because this is also something that's debated, because it has been suggested there's potentially compartmentalization of the virus. So it might be enriched in lymph nodes or in GALT or in the brain. So we have looked, and it actually looks like peripheral blood is a pretty good mirror of what goes on in the tissue. So we believe peripheral blood is a good place to to measure the virus. So the problem with the PCR-based assay is that they overestimate the amount of virus. So we know that probably in most individuals, at least 90% and maybe even 99% of the HV DNA that we measure is defect HV DNA, so it cannot give rise to an infectious variant. So we overestimate the reservoir. So there are other assays that we have to use also, which are assays like viral outgrowth assays, where we try to culture out virus of CD4 T cells. So these assays are, of course, very elegant, and you can sequence the virus and kind of describe the virus. The problem is that you don't get all the infectious, all the replication competent viruses to grow out when you stimulate. So if you stimulate once, you'll get some viruses. If you stimulate the negative wells, then you'll get some more viruses and so forth. So it underestimates the size of the reservoir. So it's really tricky to measure the reservoir. And now there are full-length assays that detects full-length viruses. So maybe this is a more accurate estimate, but really it's tricky to measure the reservoir.
You need an imaging modality where you put a patient on an imager and it shows you, you can quantify it.
Exactly. And there are people trying to do that, and it's not that easy.
No, of course not.
Yeah.
Of course not. So are you finished with TLR9 as far as the cure goes?
Not quite. So during my time of working with these latency reversing agents, I was contacted by a professor, Michel Nussensweig at the Rockefeller University in New York. And he is a really brilliant B-cell immunologist, and him and his lab had developed these monoclonal antibodies, which are called broadly neutralizing antibodies, which are basically antibodies that have been produced by cloning B-cells from these few individuals that are very good at making antibodies against HIV. And the antibodies they make neutralize not only their own virus, but a broad spectrum of viruses. So he was interested in collaborating and working with the antibodies and agents that could either reverse HIV latency or that had other kind of advantageous effects in combination with the antibodies. So right now, we're doing two different studies with some of his antibodies in patients. So one study combining the monoclonal antibodies with a drug designed, actually an HDAC inhibitor, designed to reverse latency. Mm-hmm. And another trial which combines the TLR9 agonist with monoclonal antibodies. So here the idea is, again, can we enhance immunity? Can we eliminate some of these cells that harbor latent infectious virus? And we know that the TLR9 has all these effects that supposedly, at least in theory, can enhance HCC, so all the antibody-dependent killing mechanisms. So at least in non-human monkeys, a combination of a TLR agonist with these monoclonal antibodies has been shown to induce a functional cure in some monkeys. So there's some theoretical evidence that this strategy might work, but we know it's a lot harder to cure a human than to cure a monkey or even a humanized mice. So the bar for curing a human, or at least inducing a functional cure, is very, very high.
So the HDAC inhibitors turn on transcription and make the cells make virus, and then the monoclonals would neutralize it, right?
Exactly.
And those cells would die as a consequence of virus infection.
Yes, that's
so they're gone. Hope. That's the idea. Does anyone try checkpoint inhibitors?
Yes.
As well? Do they have any promise?
So I think they have some promise. So there was the first trial that was conducted in HIV patients. The idea was again to enhance HIV-specific immunity. So unfortunately, even though the drug was the drug was given at a very low dose, I think it was about maybe 3% of the normal dose that would be administered to a cancer patient. There was one patient that had possibly this side effect of having lowered cortisol levels in blood. So there was a fear that the adrenal glands were actually damaged by this compound. And since it's healthy patients that we're enrolling in these trials, it's the level of side effects that we can accept in these trials is pretty low. So we cannot harm the patients beyond what would be reasonable and acceptable. So this is quite important in the research, but it also makes it challenging because the things that are tested in cancer just cannot be tested in this setting because it's too toxic.
Sure. So could I get your opinion on an AIDS vaccine?
Sure you can. So this has been, as you know, a concept that has been pursued for more than 30 years. I think maybe since 1984 or something. I think there's been attempts to develop a vaccine, and in the beginning everyone thought it would be quite easy to do. It has shown to be tremendously difficult to develop both a prophylactic but also a therapeutic. Vaccines prophylactic would prevent infection. Therapeutic would probably, hopefully, induce a functional cure in an individual. Both has just proven to be super difficult. So there are a lot of different concepts now in both preclinical and clinical testing. Some of them have some promise. There's the famous TWiV trial where there was a protective effect of I think around 30% of this prime boost regimen, which is now being expanded 10 years later to a trial that is ongoing in Africa. But I think right now there's some cautious optimism, but I don't think it's around the corner. We'll say within the next few years that we'll have an HV vaccine.
You know, even Jonas Salk tried to make a HIV. He said, "Oh, just inactivate the virus. That's it." It was in the 90s, right?
Yeah.
Okay. Maddy, you're almost done, right? You're writing your thesis?
Yeah, a few more weeks to go, and then I guess hopefully everything will be accepted, and then the defense before the end of this year.
What are you going to do next?
So short term, I'm lucky enough to have a position as a postdoc in Trines lab for potentially the next year or so. But after that, I'm open and looking into maybe traveling again, maybe doing a postdoc in a new country and challenging myself again and see how that would go.
So in this 1 year that you remain in the lab, what are you going to do? Are you going to continue the work you've been doing?
Yes, I think we'd like to maybe try and elucidate some of these mechanisms by which Pol3 is sensing VZV and DNA viruses, because there's still so much that we don't know. So if I could scratch at the surface of that a little more, maybe that would be nice.
If I can add, this is because Maddy has really interesting results again into what we talked about first, about what is the role of Pol3 in sensing in the cytosol versus nucleus. Maybe there's also things happening during virus infection in terms of moving in and out, and also the question of whether this is happening in the CNS or whether this is controlling latency. The Pol3 system is also something that Maddy will try to address.
Yeah.
Where are the transcripts being sensed?
They're sensed in the cytosol.
Pol3 by what? By which?
The Pol3.
No, the Pol3 transcripts are being sensed by an RNA sensor, right?
A RIG-I.
It's RIG-I.
I mean, yeah, in the cytosol.
And so the kids, they have mutations in the Pol3 gene, but not in RIG-I ever. You don't see mutations in RIG-I gene.
We haven't seen it.
It's interesting, right? Okay. Well, good luck.
Thank you.
Let me ask you all just two more questions. If you hadn't been a scientist, what would you have become, Soren?
I have no idea. I would probably be unemployed, so I cannot fit into rigorous schemes. So I would probably be in and out of jail. I don't know. I have no idea.
Maybe you would sell eyeglasses.
Yeah, maybe. Yeah, I could get a job there. Yeah, and there's also a cafe in Copenhagen called the Paludan Cafe. So maybe I could swap between.
Really?
The two. Yeah. Yeah. I don't know. I've never thought of it.
Okay. Trina?
Well, I'm already trying to fill out the two things, to be a medical doctor and to be a scientist, which is more than enough. But I think if I couldn't do these two, I think they are really perfect for me. Maybe I would be in the kind of reading, writing business, because I very much enjoy writing.
Ole?
Yeah, that's a good question. So like Trina, also have my medical doctor background too as a fallback position if research it runs out of luck or money. But as a kid, I want to be an archaeologist and dig in Egypt. So I mean, I might do that in my spare time when I get some spare time.
That's cool. How about you, Maddy?
I always thought actually being a detective is a bit like being a scientist because you're trying to figure things out. So I think something maybe detecting or a policewoman or something like that. I always like the idea of being in a more maybe action job.
You watch too much, what is it, The Bridge?
Yeah.
So RNA or DNA?
Oh, DNA.
RNA or DNA?
DNA.
Trina?
DNA.
DNA, I guess, for me too.
It's got we got a little mixture here. All right. So that is TWiV from Aarhus. Is that how you say it? Aarhus. You can find TWiV at microbe.tv/TWiV. If you listen on your phone or tablet, please subscribe so we know how many people are listening. And if you have questions or comments, you can send them to TWiV at microbe.tv. And if you really like what we do, you can consider supporting us financially. Microbe.tv/contribute. We have PayPal and Patreon accounts where you could give us $1 a month or whatever your currency is. And we do this on our own with not much other support, so that would really be appreciated. My guest today from Aarhus University in Denmark, Søren Paludan, thank you for joining us.
It was a pleasure.
Trine Mogensen, thank you very much. Ole Søgaard, thank you very much. I'm sorry about the name, but thank you for joining us.
Perfect, and thank you for trying so hard to pronounce my name.
And Madalina Carter-Timofte. Maddy, thank you, and for inviting me to come here, taking me to dinner last night. Appreciate it all. Walk a little bit around Aarhus. So thank you so much. I want to thank ASV and ASM for their support and Ronald Jenkins for his music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Thank you.
Yeah.
Thank you.